Alrighty, this is a brief introduction to DNA technologies. And DNA technologies are basically the different types of techniques and technology that we use in order to look at, identify, and manipulate DNA. Alright, we're going to start off talking about cloning. Cloning is something that most people have heard of already. You might think about examples when you see movies like Multiplicity with Michael Keaton, or Austin Powers with Dr. Evil and Mini-Me, his little clone, or even Splice. You know, those are a few movies that kind of talk about or show examples of cloning. On a long story short, what cloning is when you take the DNA of one organism and you put it into another organism in order to create the exact same organism. You know, kind of just like another copy. Almost if you were to put a sheet of paper in a copy machine and then press the copy button, it would make an exact copy of that. So pretty much the same kind of idea. Researchers clone animals by nuclear transplantation. And that's basically, in a nutshell, when a nucleus of an egg cell is replaced with the nucleus of a somatic cell of an adult organism. And we know that a somatic cell is any type of cell that's not an egg or a sperm. Okay, so anything but a sex cell. And to answer the question, do we clone humans? Can we clone humans? Thus far, attempts at human cloning have not succeeded in producing an embryo of more than 6 cells. So, in order to, create a human, you need a lot more than 6 cells. So basically, no, we cannot clone humans, and there are other ethical issues with that as well. And by ethical, I mean a lot of people think that we should not because of some reasons that they believe, and just kind of it's a very controversial topic. So let's move on. Alright, so in the early cloning situations, we use frogs. And on the right, you have a display or diagram kind of showing how they would do it. The cloning of tadpoles showed that the nuclei of differentiated animal cells retain their full genetic potential, which means that the cloned organism has the full genetic makeup of that organism. It has all the DNA to make another one or to make another one, so that they're not limited or lacking anything. Alright, with recombinant DNA technology, researchers basically kind of studied E. coli, which is a type of bacteria, and it revealed that these bacteria have like a sexual mechanism that allows them to kind of combine genes from 2 different cells. So this discovery led to the development of recombinant DNA technology, which is a set of techniques for combining genes from different sources. So basically combining genes from different organisms. Okay. Alright, I'm going to talk to you about this, and then I'm going to zoom in on this diagram to the right so that you can see exactly how it works. And I mean this diagram over here on the right. Alright, so first, we're talking about genetically modified foods. You might kind of question, what is genetically modified foods? Okay, pretty much, these actually are more common than you think. More than likely, everyone has probably eaten some type of genetically modified food. So here's the example. Well, I'm going to tell you in a nutshell what a genetically modified food is. It's any food that has been, I hate to say tampered with, but any food that has been changed or had something added to it in any type of way. For example, if you see, if you have grapes that don't have seeds, that's a genetically modified food. Most fruits, that don't have seeds in them have been genetically modified in some type of way. If you ever see a exotic type of fruit, like a blue strawberry or something like that, that could be a genetically modified food. But let's go ahead and get into the details. So when you do this, you insert beneficial genes into the plant/organism's coding sequence. Alright, so basically you're inserting pieces of DNA inside of the DNA of whatever organism you want to have those traits. So the example they give is a crop is grown up having these genes. All organisms in this crop will have the specified resistance / benefit. Oh, I guess I need to explain that first. So let's go ahead and zoom in on the picture over on the right. Alright, so you see the DNA right here of some type of beneficial gene that you want. Let's say a guy is a farmer and he is growing corn, and he has these insects that keep eating all the corn before it gets to be fully matured and grown so he can pick it. So basically he's losing a whole lot of money and a whole lot of time because these insects keep eating his corn, and he needs to find a way to stop these insects from eating his corn. And the insects never ever bother the carrots. Okay, so he basically would find what in the carrots that the insects don't like, or whatever in those carrots that repels the insects, and he would take that out. He would find it in that DNA of the carrot, which it could be kind of, let me find the cursor right here. So this little part right here, this could be the BT gene, which is like a resistant gene. It makes this gene right here is resistant to those insects that are eating the crops. So you would use enzymes, restriction enzymes, to pretty much separate the DNA, take out this part that you want. You would take that part out that has the BT gene, and then you would insert it into the corn's expression cassette, which is basically saying insert it into this part of the DNA of the corn. And then you would take that and you would grow it in bacteria. So it says vector with transgene multiplied in bacteria. So you would take that BT gene you wanted, that resistant gene, and put it into some DNA, and you would let the bacteria basically you would insert it into the bacteria would grow, and it would grow more and more and more and more of those cells with that resistant gene. Then you use a plasmid and you insert it into the actual nucleus, into the genes of the corn cell that you want. Alright? And then once you do that, you have corn that's growing, and now that corn has the resistant gene in it down here. Alright, so basically it's just a way of finding a trait that you like and you want another organism to have, using restriction enzymes to cut it out. You can take it, insert it into that gene, so you make this strand right here, and then you use bacteria to, allow bacteria, or put it into bacteria, and then bacteria will grow it and grow multiple copies of it. Then once you have that, you use a plasmid and you insert it into the DNA of that corn, and then that corn would grow and it would produce the corn that has that gene inside of it. Alright, give me 1 second to zoom back out and get rid of this magnifier. Alright, here we go. So I talked earlier about those restriction enzymes. What a restriction enzyme does is it's used to cut DNA fragments. Okay, well, it's used to cut DNA into fragments. If you saw earlier when I showed you with the cursor how that BT gene that we wanted, we used the restriction enzymes to cut that DNA out. So that's what restriction enzymes do. They cut DNAs into fragments. And it says they can either remain into fragments for sorting via gel electrophoresis, which is another type of DNA technology we're going to talk about in a second, or they can be inserted into a plasmid for future implantation into a bacteria for protein proliferation. Long story short, they can either be inserted into a plasmid, just like that example that we just talked about, and then put into the bacteria for future use, or they can remain in the fragments that are cut up, and you can use those in the gel electrophoresis process. Alright, here is the gel electrophoresis. Long story short, this is how it works. You take fragments of DNA and you insert them into this gel. The gel kind of looks like this down over here. You insert them into these little slits right here of the gel, and then you actually put a positive charge and a negative charge over here, and you basically apply electricity to this gel, and then the DNA will pretty much run its course, and then at the end it'll produce a picture which will allow you to kind of compare DNA. Alright, and on the next slide, here we go. This is what the picture will end up looking like down here on the right, where you have these different slots, and then you can compare different organisms' DNA and see how closely related they are. Or if you have an unknown organism's DNA, you can match it up and see whose DNA it is. Okay, so kind of like if you had a crime scene or a paternity test and you wanted to find out if the father of the baby is this person, kind of like a Maury. Alright, and this is called DNA fingerprinting. So this thing's over here, this is your DNA fingerprint, not like the ones on your thumb. Alright, so you use gel electrophoresis in order to identify individuals. It says no two people besides identical twins will have the same DNA fingerprint. So DNA analysis can be used to help solve crimes. And it's talking about, remember, these fingerprints, not the fingerprints on your fingers. Alright, next we're going to talk about gene therapy. Alright, it says gene therapy may someday help treat a variety of diseases. And it's a technique for manipulating DNA that has potential for treating diseases. You're altering an afflicted individual's genes. So basically, it's like if a individual had cancer or something like that, maybe, and hopefully in the future you'll be able to pretty much modify or manipulate his DNA by taking the DNA from something else and injecting it into the individual in a certain way, and then hopefully producing better cells or curing whatever needed to be cured. I'll kind of go through the example again when I zoom in to the right. So here you have a cloned gene. It's a normal allele. So this is a cloned, remember we talked about cloning? This is a cloned gene, so it's not a natural gene. It's cloned, but it's the normal cells. And if you would put it into someone that has maybe bone marrow issues where they have bad bone marrow in their bones and they need more cells, more normal cells, you would take that gene, the normal gene, you would insert the normal gene into a virus, okay, so into a viral nucleic acid, and then use a retrovirus, which is like pretty much a virus that you kind of basically change, or modify by inserting this little, where's it at? Let me get the cursor. By inserting this little, piece of the DNA, and then you inject the bone marrow with the virus, and then the viral DNA is inserted into the chromosomes, which is right there, and then the bone marrow cells from the patient. And then you inject the patient with the bone marrow, and then it's supposed to grow inside the bone marrow and hopefully create more normal cells. Alright, so let me get back to where we need to be. Alright. And lastly, the Human Genome Project. The Human Genome Project involves genetic and physical mapping of chromosomes. And by mapping of chromosomes, I mean it's pretty much kind of analyzing chromosomes to find out how to make the copies of them, what they do, how they do what they do. DNA sequencing, which is pretty much when you start to find the matches and copying of copying of DNA. The comparison of human genes with those of other species. And long story short, the Human Genome Project is a project that we're trying to get the full genome of a human, meaning all the chromosomes, all the DNA in a human, and trying to analyze each of those chromosomes and find out how they're, and genes, how they're made, not made, but what the base pairs are, and pretty much kind of how it works, so that later on, if we need to, we could make copies of genes that we need or things like that. Okay.